Welcome to our third lecture this semester in AgScience-on-Tap. It's a rather small crowd. I think it's because it's not raining today, but it's going to be an exciting event, I think, because it's going everything about chocolate, which it's one of my favorite foods. So the topic for the semester is sustainable food production and nutrition. We already had listened to two talks, one about alternative proteins. We have the speaker here, so if there are still questions open, you can still discuss with her. Then last week we heard about something about healthy people on a healthy planet. And today we're going to be hearing about how chocolate will taste in the year 2100. So let's see what climate change is doing to my favorite food here. I welcome Andreas Dunkel. He's the head of the research group for integrative food systems analysis at the Leibniz Centrum for Food Systems Biology. And he's also co-chair of the Institute's database facility. And I don't exactly know what you're doing, so I expect you to introduce a little bit on your work. So his research focuses on developing workflows for the comprehensive characterization of complex food systems. And he used mass spectrometry and data science approaches. And the aim is to understand the impact of new material composition, processing parameters, and storage conditions in order to optimize the hedonic value of food products. Is that correct? Perfect. So the floor is yours. I'm excited to hear.
No? Yeah. Okay, dann habe ich es jetzt entmutet. Well, okay, so thanks for the kind introduction. And actually, I will try to show you a little bit of a story how somebody who started to work on taste and developing analytical methods as a traditional classical food chemist, how a person like me ended up in doing climate change research and thinking about how certain products will taste in the far future. And so I will include a little bit of science in between to show you also a little bit of the complexity of what we are doing and how difficult it is in some cases to work on these topics. Okay.
The loudspeaker, you mean, huh?
Yeah. But I will try to keep it simple and give you more or less the ideas how we use these complex techniques to come to the results. And at the very end, I will do a small experiment with you because we prepared some, let's call it chocolates. So it's not exactly what you would buy in a supermarket, but it resembles all the ingredients that a chocolate has. But in one example, we did a small change that hopefully gives you an idea what actually our biggest fear for the future of nice tasting chocolate. But that will be more at the end of the presentation. So let's start first with some introductions. And one very recent development is that we have a real severe spike in cocoa prices, and chocolate prices will be up as well in the near future. So, for example, yesterday, Spiegel Online, a big German news magazine, published an article which says in the end, the world of chocolate will never be the same. And this is due to many of the developments which are primarily due to climate change, but also other effects which are due to the economic situation in certain countries of origin, especially in West Africa, will have also a major impact on the prices of cocoa. So this is not only something you find nowadays in general news. You also see this, for example, at JP Morgan, which is one of the largest financial service providers in the US, which also looks at the topic and published an article in April, which says, is this leading into a real chocolate crisis that we have heavily increasing cocoa prices? And what will be the future development? So how will the market of chocolate producing companies will change? And also Bloomberg, so another important provider of information, especially about economic data, showed, for example, in April, that the current price of cocoa on the world market is more than double what we found any time in history. So the former spike was here a long time ago, more than 40 years ago, where we were topping the price at about $6,000. And now we are well above $10,000 currently. And there is not really an indication that this price will go down again significantly. So this already gives you a first idea why it is so important to think about the future of cocoa production and chocolate production. And that's also why research now really is starting to increase dramatically, because there seems to be a real need in the market to change the situation. So let's look a little bit deeper at what we do and how we can use our research here in Freising to look at this topic. And I just want to show you one picture about our institute to explain a little bit how research at our institute is constructed, because we try to look at food systems in general. So we not just try to understand, for example, what's the molecular composition, which are the ingredients of certain foods, because this is more or less just one part of the game. We also need to know on a fundamental level how compounds interact, for example, with receptors in our body to trigger certain sensory events. And we also need, and that's the research section where I primarily work, then to predict and integrate all these data. So heavily using new developments like AI-driven prediction methods and so on. And this is on the one hand contributing to, of course, the production of healthy products. But all healthy products that we have in mind in the development currently are not successful very likely on the market if we are not also think about the sensory quality. So even if your doctor says to you that's something which is very good for your health, but if you don't like the taste, it's very unlikely that you will consume it for a long time and frequently. And therefore, we think only if these things play together, we are having a chance to transform the food market and develop new successful products. But these products, of course, need to be also sustainable. If we need lots of energy to produce certain crops, for example, then it will be again an issue if we think about conditions in the next decades where we definitely need to be where we need to work on a reduced carbon dioxide footprint and all these things. So sustainability of the food production is as well becoming an important research pillar in our institute. And as we work on the integrative analysis of such food systems, we think that's the best way to really tackle it from all different angles. So how can we analyze such a food system, especially the traditional way to understand, for example, the taste? Well, that's on the one hand a very simple picture, as you see here on this slide. In real life, this is actually at least a PhD thesis usually, because you need to really step by step follow the different steps here on the slide. And if we start with a complex food system, like on the left-hand side, first step is identifying the molecules which are, for example, behind the taste and smell of a product. And only when we have achieved this list of molecules, which is really relevant for a specific product, we can go on then to develop, for example, methods to quantify those compounds in a high throughput manner. So that means we are then able to very efficiently monitor, for example, the changes of different important compounds between different varieties of a crop, or also what happens, for example, during processing. And then we can use this to build certain models. For example, what happens if you change a certain compound in the complex mixture for the taste of, for example, an orange juice? Is it getting more acidic? Is it getting more bitter? Because only if this is still in balance, you will have a product which tastes nice to you. Cannot simply add, for example, more sugar and expect, oh, everybody will like it afterwards. Because if there's a certain level where this will shift, and then, for example, the complex profile of orange juice, which also requires some bitterness and some acidity, will get lost if you have too much sweetness. And all this together is then enabling us to do prediction, which means we can, in the end, understand what will happen if the food system changes in a specific way on the taste of the product. So that's the basic process, but of course it's much more complicated, and at the different steps, lots of different techniques in the lab are needed. So let's go to cocoa, and let's have a very simple look at the compounds which you need to get a nice chocolate or especially cocoa taste. So if you think about the taste of cocoa, it's different than chocolate because cocoa, of course, contains no sugar. So everything you will perceive is bitterness, astringency, and acidity. So I think everybody knows bitter from home when you drink your coffee and so on. Astringency is what you perceive usually if you eat certain foods. For example, if you drink a red wine, a dry red wine, or if you have a black tea, then you have this kind of puckering, rough feeling on the top of your tongue. And this is called astringency. And this is caused by many plant compounds, like, for example, flavonoids, like on the top. So that's something you need at a certain amount in foods. But if it gets too much, people also again start to dislike the products, because you will have then for a very long time this kind of furry feeling on the top of your tongue. And this is disliked by consumers, of course, if it happens too much. And also the acidity, as I already mentioned, is a part. So if you want to understand how cocoa tastes, you can more or less taste the traditional baking powder you have at home. The only thing that is different to the taste of full cocoa is the acidity, because the baking powder you can buy is already alkalized, which means the acidity is kind of buffered in it. But you clearly get the bitterness and astringency you see in the cocoa. And here you see just a selection of the compound classes which are responsible for these compounds. And some years ago, we were tasked by some industry partners to develop methods to help them to quantify these compounds along their value chain very efficiently, because these are very specific compounds, so you need high-end analytics usually to quantify them. And that's usually not what you see in a typical food manufacturing environment. There you need usually more simple, more efficient and faster methods. And I will not go into the technical details, but we developed a quick method which is able to work with all materials from beans to final chocolate, and getting then from our complex machines, so the mass spec instruments, chromatograms like that, and all these different signals are in the end indicators for certain taste compounds, and we can then, with the help of that, predict, for example, how much a certain variety or a certain processing step contains, and can predict based on that the taste of a product. So we use this then in the next step to build the first kind of word map of cocoa taste. So we gathered from the word market lots of different samples and use then our new methodology to screen them, to understand the distribution of certain compounds like the theobromine and the caffeine, for example, for the bitterness, or certain astringent compounds, to give the industry for the first time a very rough picture. What's the distribution and different origins? So how cocoa from, for example, South America differs from West Africa, which is the main producing area, and especially how different it is from new regions which are coming up now from Asia, for example. So this is something which was already a big help for the industry, because they could now, with a very quick method, already use screen materials they want to buy, because for industry it's quite difficult so far. They just buy almost a whole ship of raw cocoa, and then they have to do tests to evaluate, is this something they can use for a really high quality product, or is this maybe of poor quality, which you can just blend into certain products with probably a lot of sugar and so on, where the taste of the basic cocoa is not that important. But especially when you work, let's say, on pralines or chocolate with a high chocolate content or a high liquor content, let's say 70–80% of cocoa, then it's really key that you have a high quality cocoa. And selecting these materials is really something where these methods were a big help. And actually, this is still traditional food chemistry, so this was what we do in our lab all the time for different products. And at that time, we were not really thinking about doing more research, especially we were not thinking about climate change at that time. But then during Corona, I gave a talk about these methods, and afterwards I was contacted by some researchers from England, so probably another country which you probably don't associate with cocoa research, but actually it's one of the most important. And they asked me whether we could work together, because they are already for decades looking into diversity of the cocoa plant, and especially also into disease management, and they recently started also to work on climate change. And these partners are working at the University of Reading in England, and they are running since the mid-1980s already a cocoa research center and also the International Cocoa Quarantine Center. And this is the only place worldwide, more or less, where they try to control what happens if you transfer a new clone, for example, from South America to Asia. Because the problem is if you just bring the plant there and try to breed it there, you have a high risk of transporting also different viruses and so on to these locations, which currently don't have issues with these diseases. And therefore, the idea of the cocoa industry who is funding this was in the 1980s, yeah, let's build a quarantine center where the plants can be housed for several years before they can then, after lots of testing, be transferred to the new place of origin where then the breeding can continue. And this place, of course, now hosts lots of many different genomes of cocoa and also clones. So currently there are at about 400 different clones, for example, the cocoa world. So it's one of the largest collections worldwide. And of course, if you have such a large diversity at hand, you also start to think about doing some research and not using this as a kind of service for the industry. And that's what they started to do some years ago in their greenhouses, that they tried to look at climate change. And this was initiated by climate models, which were calculated by another colleague from the University of Reading, the Emily Black, published this paper a few years ago. And they looked at what will happen very likely at about mid of the century and end of the century in terms of climate in West Africa. So West Africa, that's a region primarily composed of Ghana, Ivory Coast. Those countries are primarily important for cocoa production. And all this is based on simulations, but those simulations nowadays are quite good, as we have to learn every time we have a heavy flooding like currently. And those models, for example, show that we have to expect at the end of this century in West Africa different things. So on the one hand, we have to predict an increase in rainfall. So you might think, oh, everybody expects it's getting more dry. But actually, what the models predict is an increase in rainfall, but the problem is here on the right-hand side, that we will have more severe kind of seasonality in the rainfall, which means we will have longer periods where it's very dry, and then we will have very short, extremely heavy raining seasons. So that means this will be then completely different for a plant which expect to have more humid conditions for most of the growing time. And if it gets into dry and then just a lot of water in a very short time, it's of course an issue, and we have to learn how to deal with that. The other point they identified was, and that's of course also not very surprising, they are expecting a temperature increase. And that's of course not only something we expect in West Africa, but in this specific region, these models say we will have about 5 degrees C increase by the end of the century. So 2.5 degree at about 2050, and at 2100, we really expect in that region about 5 degrees of increase. And this will go together, of course, also with a global rise in the carbon dioxide, the atmospheric carbon dioxide level. And this was something they tried to understand to predict also what will be the effect of those conditions on cocoa productivity. So will this go down? Will it maybe go up? And so they started to do different models. And the basic experiment I want to show you was done in their greenhouses and climate changes by just simulating those conditions. And I will skip those parts which look at the precipitation and the rainfall and just focus on the carbon dioxide and the temperatures. And what they did, they used a full factorial design, which means they combined different levels of temperature, which present current date, 2050 and 2100 in the last level. And this was combined then with different carbon dioxide levels, so 400 ppm in the atmosphere, that's what we have about right now, and 700 ppm, so almost doubling it, is what we expect again for 2100. And as cocoa is a quite diverse plant, we also need to look at different genotypes. And for the initial part of the experiment, we started with 2 important genotypes, the so-called CCN51, which you can kind of translate to something which is called bulk cocoa. So that's not the highest quality of cocoa, but that's used in many products where cocoa is just a part of the product, where it's probably just added on top as a little bit of chocolate, things like that. And then we use a second variety, the Scalina 6. That's a fine flavor cocoa. So this is something you want to use for high quality products where you have not many other ingredients, where really the cocoa needs to stand for itself and has a high quality. And those trays were planted in the greenhouses, grown for several years until they were able to harvest something. And then you get these small pots, and those pots can then be opened, and you can then get the cocoa beans out of it. And this was in the end analyzed first by the group there, and they are plant science people, so I have no idea about all their methods, but they looked at many different effects, like, for example, photosynthesis. They look at the simple weight of the plant, so how much is produced, for example. And they were seeing some quite interesting effects. So the temperature increase was something they were not really expecting at that level, but they were clearly seeing that it has an effect on photosynthesis, on vegetative growth, on the pod and bean characteristics. So, for example, how thick is the shell of the cocoa beans? All these parameters, they were analyzed by the people there, and they tried to understand, is, for example, a temperature increase or a carbon dioxide increase having an effect on those parameters? And surprisingly, they were seeing, of course, the increasing temperature has a negative effect, but several of the effects were kind of compensated by the increasing carbon dioxide level in the atmosphere. So we, of course, think carbon dioxide increase in the atmosphere is something bad because it leads to the increased temperature levels and so on. But of course, a plant is doing photosynthesis, and if there's more carbon dioxide, that's not totally bad for a plant in certain ranges. And therefore, what they were seeing, depending on the genotype, that, so for example, if you take here the light gray bars for this genotype, if you look at increasing temperature, the productivity really falls down. But if you keep the carbon dioxide up, you see the drop is not that severe, and the productivity seems to be maintained at a certain level. So that looks not that bad, and that was definitely not what they expected. They thought it will have a much bigger effect on the cocoa beans and especially the overall productivity. And so their claim was in the end that they predict the overall net productivity in West Africa, which is the main cocoa growing region, will not be significantly lower at the end of the century. But the big question was, and that's why they contacted us in the end, will there be an effect on the quality of the beans? Because they were already seeing, yeah, the cocoa bean husk, so the shell around gets thicker and so on. So this was already an indication something changes in the plant. And the question is now, will this also have an effect on the sensory? Because nobody will be happy if we produce still cocoa, but it tastes awfully and nobody wants to buy it, because then it's also not great. So this was something they want to know, and that was when we started to collaborate with them about 2.5 years ago. And the first thing was that we were thinking, is it really a good idea to use a methodology that we just published to make this cocoa draft map that I already shown to you? And we were thinking about, yeah, that's probably a little bit too simple if you just take a picture. And I want to show you why we were not doing this with just 2 simple pictures. Because you might say, okay, we use all the tastings in this method. Let's monitor them, and let's try to understand, is there an effect on those tastings or not? But we were thinking about, is this probably not enough of the whole picture, and do we need to monitor the whole picture? If the entire plant, so to understand really what happens in the plant, or so if it's not important for the quality of the compound, of the taste of the product. And therefore we were using so-called holistic untargeted methods, and that's what you'll see here on this slide, to just show you how important it is to not just look at a few compounds. It's really important to see the whole picture in many cases, because I would expect this pool looks much more interesting than that one, although the other part of the environment might look nice to you. So this really shows you how we have to select the method. Sometimes this is sufficient for certain questions we are asked in our research, but in other cases where we want to understand how the whole system changes, what really happens in the plant on the systems level, we need this picture to get really the overall view. And this was what we were doing in the lab, and I will not spoil you with all these technical details, so we will just jump over it. But in the end, we use lots of different machines to acquire lots of data, and that's what also tells you what's nowadays in our research an equally important part. It's not only having these machines and acquiring lots of data, you also need to analyze the data, and in the end, that's what we do in our research group, that we really have experimental work, but we need to have also lots of programming and data analysis work, because otherwise you cannot find the interesting points in these complex data sets. And We will not go deeper into this, but I just want to give you an impression how complex these methodologies are. And if you just look, for example, at such a cocoa sample that we analyze, you get then from a machine such a map, and each of these 10,000 of points is one different molecule. And that map you get for each different sample. Then you need to do, of course, lots of statistical analysis to find actually which compound is which of these points and which of those is actually important and which are those which are not really changing under the different conditions we're analyzing. And we can then, with our tools, go into these different points and identify then certain molecules, like, for example, these procyanidins, which are important for the astringency, and then really analyze and compare it also with libraries to really be sure that's exactly the compound that you can do with all these different dots on this map. So that helps us to first identify the compounds, and then we can use statistical tools to understand which of these points is important. And I will just show you some of these maps to give you an impression what happens under these conditions. So as I explained to you, we have different genotypes, we have different temperature levels, we have different carbon dioxide levels, and all these effects, of course, overlay. So it's not a simple separation. Here is one temperature level, and here is the other temperature level, because the effects between different temperatures are overlaid, for example, by the different genotypes, as you have seen also by different carbon dioxide levels. But the major points we were seeing in the data, first of all, the differences between the genotypes was definitely larger than all the temperature effects we were seeing. And then second to that was the temperature effect. But we were also seeing, for example, a small increase in temperature for mimicking the conditions of 2050 was still not that different from the conditions we have nowadays, and only the conditions we see in 2100 will be severely different. But that's just part of the picture, because if we look a little bit closer at the different genotypes, so this was the fine flavor cocoa, and this was the bulk cocoa I told you, then we already see a difference. While this bulk cocoa, so that's what we do not really like, because it's just a basic plain cocoa without a nice flavor. This seems to be much more robust. So at least the 2.5 degrees increase we expect for 2050, this seems to be compensated by the plant somewhat. And only then at the levels we see in 2100 or predict for 2100, we see a huge change in terms of composition. The fine flavor cocoa seems to be much more susceptible. So that already is showing heavy changes at the 2050 levels. So there we need to act much faster to find a solution compared to the bulk cocoa. But as in most cases, that's what we want to have, although it's probably not the best and most robust product. So we also can, of course, then go deeper and look at different carbon dioxide levels and how this affects us. So the overall message is we have tools to detect on the molecular level what makes these different cocoa beans different. And then we can use this to understand which of the many different compounds in it are responsible for that, to understand what happens in the plant, and also how might this affect in the end the taste. And I will show you with another picture how we go then to the real individual molecules from this large map with lots of green dots you had seen. Therefore, we use, for example, different libraries to compare those compounds with the data we acquire with our instruments, and then you can use this, for example, to compare to the libraries and understand that's exactly the compound I'm looking for, and that's something which is, for example, different. And to integrate all this knowledge, we are then using network analysis, and this is giving you a picture like that, and each part of this network is in the end representing a compound class. So everything which is clustered together here in such a small subset of the network is heavily linked by a certain similarity in terms of chemical structure. So that means things which are in different groups here are structurally different. And then we can overlay this, for example, here for the temperature effect to see is a whole class moving into the same direction in temperature increase or temperature decline, or is, for example, like in this compound class here, one half going in one direction, and the other is in the other direction. So this helps us on a very global level to understand what happens when we change the temperature. And I just will show you for one small example of this map, what happens in detail on individual compounds. So let's have a small look at some compounds. So for example, here you see something from the polyphenol chemistry, or the trigonylene or something. Here we see, for example, plant hormone, and these are now data for individual compounds for exactly the same conditions. And we see, for example, here nicely for this plant hormone, at the first two levels of temperature, they are the same, but the plant hormone is even increasing at higher temperature. And this is then something which seems to be related, for example, to the development of these thicker shells of the bean, which is not necessarily something we want to have. And on other ends, we are, for example, seeing a decline of compounds which are usually something you want to have because these are compounds which are nice precursor for the cocoa flavor. So that means this is a compound we need that we get after we roast finally the cocoa, a nice smell. So in the raw beans, this is not interesting. But if it's not in there, you will not get after roasting something nicely smelling. And that's why we also need to look at those compounds which we call precursors, which are just in the bean because they will be more important later in the process, but not directly already at that level. So that means we can look at different compounds. And I mentioned already, we can, for example, go then here in such a small part of the map. And we see then here compounds like the trimethyluric acid. This is a xanthine derivative, so this is very closely structurally related to compounds like, for example, caffeine or the theobromine, which is one of the key tasters in cocoa. And what we clearly see with temperature, this of course goes up, but the effect is very different between the genotypes again. And this tells you already, okay, there seems to happen a lot when we have different conditions in the near future, but it's not necessarily with every genotype. And maybe selecting different genotypes for certain parts of the production regions might be an important step to have at least until we reach other points in the climate development, still a product which is kind of giving us a nice flavor and enough production. So when we go on these maps, as you see, we really can then zoom into it and see all these different structures. And if you really see then that all structures which are such nicely related to each other, this is something telling you, okay, the system, the entire plant works in a certain direction. And that's not just something like noise. So one compound goes in that direction, that goes in another one goes in another direction. This is really something integrated and showing a systemic response by the plant to certain external developments like the climate change conditions, for example. The other effect we were interested in was understanding how does the cocoa fat changes, because usually when you talk about chocolate, a huge part of it is not just the cocoa as you know it, it's also the cocoa butter. And that's a very important and very fragile product. So the cocoa butter is extremely well suited to what we use it for. So cocoa butter melts exactly at your body temperature, a little bit above. And that's very important because if it's not happening, then the chocolate will not melt on your tongue and you have something very grainy. So it's more like chewing on a stone and not getting nicely dissolved in your tongue and that you're able to swallow. So that shows already parameters of the cocoa butter are quite important. And to understand whether there are changes as well when we have a climate change effects, we looked also into the complex lipidome, which means the entire lipid compounds we see in such a sample. And this is different to former times where we just looked at free fatty acids in such a sample. Nowadays, with those machines, we can really look into the complex triglycerides, how certain fatty acids are attached in different phosphopeptides and so on, so that you get really maps like that again, which are then clustered into different compound classes. And this was what we were analyzing as well in our samples. And just to give you a short example here for two compounds of example, here you see one of the fatty acid, the stearic acid, which is a saturated acid, which means it has no free double bonds. So it's a very rigid fatty acid. And actually, if you reduce the number of double bonds, this is leading to a fat which has a higher melting point. And as you see here, there seems to be an increase in the level of this compound, for example, here in the year 2100 conditions. While on the other hand, if we look at complex phospholipids with high unsaturation, so where we have many of these double bonds, which reduce the melting points, we see this seems to go at the same time down. So we have on the one hand something increasing that we don't want to have that high. On the other hand, the things we are interested in go down. And this is something which we are really worried about because this might in the end result into a cocoa butter which melts at much higher temperatures only. So that you probably only will enjoy your chocolate anymore if you have a high fever or so. That's probably, then again, not the best time to start eating chocolate. So that's again something we need to look at. And luckily, there is also already research in this problem, because on the other hand, people were already for decades trying to develop products that you can sell, for example, in more warm regions, though, like in the like in Africa again. There currently chocolate is not really a product, because it would of course melt all the time if you store it somewhere. Therefore, they try to develop specific chocolates which are more suited to these temperature conditions. And actually, we currently now see that this research might help in the future to understand how we can, for example, mix in different fats. It's not anymore a chocolate under the German regulations or European regulations, but maybe we have to be open to topics like that, that we mix in other fats from other plants in the future to keep it still melting at our tongue. And I will try to demonstrate this effect later to you in our small sensory test. So if you summarize these experiments, what do we see? So we can clearly say that we can map all these different conditions, especially that are caused by the increased temperature and by the different carbon dioxide levels. And it's primarily leading to an increase in certain compounds we don't like, because they are very bitter and very astringent. So that's something which we usually want to keep under certain levels, because otherwise the chocolate will either require lots of sugar, or it will definitely not taste anymore like a nice chocolate. And especially the bitter quality is important. So it's not only the intensity, how bitter it is, it's also the bitter quality. Everybody knows that there are certain bitter foods that you like. So for example, beer, like in Bavaria, the Pilsner beers, many people like. Also, if you have a bitter chocolate or a coffee, that's products which are bitter, but they still say, oh, that's okay for me. But then there are many plants which are heavily bitter, used primarily in pharmaceutics, for example. And these are things you definitely don't want to have. So it's not only the intensity, it's also the quality of the bitterness. And if now these patterns change too much, this might happen as well. It's not only then the intensity which goes up, it might also be a change in quality. And that's then not something you'll be able to mask anymore with just adding more sugar, for example. Then still the product will taste awfully, although you added lots of sugar to try to mask it. So that's the one change we see. And the other, as I already mentioned, is that we really see for the conditions that we predict, that we expect for the end of the century, that the so-called cocoa butter hardness will increase, so that we have not a melting nice chocolate anymore in our mouth. And this is, of course, lots of data analysis and also instrumental analysis, but we are still not that far that we can just use such data to predict this is now happening. We also need to do validation experiments, of course, in the lab. And this is still requiring what you see on the right-hand side in our lab. We need still our colleagues, which are trained on doing these sensory analysis, to test the samples and to tell us, yeah, that's very bitter, that's not so bitter, and all these things. So, of course, we have these expensive machines, and they do a nice part of the job, but we are still not at that level nowadays that we can say, just based on the data analysis, what will happen. We still need to prove it. That's where we have then the people in our lab sitting in these booths and doing then the evaluation. And to not just talk about it, I want to do such a test, not under the same conditions with different booths and so on with you. Therefore, I brought just 2 samples for you. So I would suggest that first everybody takes 2 of these beakers, because otherwise it's difficult to, I don't want to have you putting the chocolate on the tables. Yeah, I'm trained, as you see, because that's what I do in every lecture. So, and I will give you 2 samples, so don't mix them up. So that's chocolate we just produced yesterday in the lab. And first thing you should do is smelling, of course. So does it? Yeah, just does it smell like chocolate? Yeah. Okay, not convinced. That's chocolate. It also looks kind of like chocolate, or? So now let's see about the taste. So I would now say take one of these, or two if you want, in the first beaker, and then give it to the next person. And then I have a second box, and this is produced from the same cocoa. We just added a little bit to mimic the changes in the cocoa butter. And I will then explain you how we should taste it. So that's the second group that we will taste in the second step. Yeah, you will not see a difference in smell. It's the same smell. It's just a small change we did. So if I say chocolate, so this is just cocoa, which means we have the brown cocoa mass, or cocoa liquor as it is called. Then we have some cocoa butter. And to make it a little bit easier for you, we also added some sugar and some milk powder. So usually it's tested without these additives, but then it's of course very bitter, and it's something very likely you would not prefer. And this is also still not at the sugar level, for example, of a commercial chocolate. And if somebody wants to taste the version almost without sugar, I also have it there. So everybody can do afterwards as well. So I will share also these boxes for you. So now how we taste it. So I would not taste the whole block at once because that's probably a little bit too much. I would probably bite out half of it for the first try. And start with the one I gave you first, and don't directly start to chew it and swallow it. Just put it on your tongue and just let it there. Let the saliva help to dissolve it a little bit, and also try to see how fast does it dissolving, and can then maybe simulate some chewing in some movement with your tongue and so on. Just pressing it then against your oral cavity. But I would not directly chew it and swallow it, because then it's probably too fast that it goes away as you are not trained on the effect already. So just take the first one, maybe half of the block. Yeah, start with the first one. And just imagine remember primarily, because you need to compare it to the second one afterwards. What happens on your tongue? How does it feel? Does it melt nicely? Or does it take forever to go away? All these things. So there's one which is labeled C, that's the regular one, and D is then the one where we did some changes. So after you had the first one, you can try the second one. Exactly the same way, and compare it a little bit. What happens on your tongue? Does it feel different? And I hope you will see a difference. Yeah, that's a difficulty when you do chocolate tasting. You cannot do many in a row because but exactly that's what we want to see, the difference, because that's what you see. Okay, somebody has already a difference. Just a little bit. That's a problem. You need to standardize it. That's why we use these labs with very controlled conditions, where we always have the same colored lights, where you always have same temperatures and so on, because you need to standardize it heavily, because we are very different every day, and that would change the evaluation every day. So, do you feel a difference?
Mm-hmm. I would say D melts quicker.
So what you also can probably, so the bitterness might also slightly change, but the primary effect is really this difference in melting. And what you probably also feel in the difference is that it stays longer on the tongue, and it creates a kind of feeling that I don't think is that nice. So it's kind of persistent there, and really feel that's so fatty. And I would say if you eat multiple pieces of that, It will be then like you would put your tongue into a piece of butter or so. Although you definitely want to taste the one with less sugar, I see. Yeah, you can. I have a whole box there for you after the talk. Yeah, no, that's the problem with chocolate. With sugar, you can still mask a lot. There you feel the difference even better because you focus really on this quality. That's also what the producers in the sweet industry need to learn. For example, which chocolate or which cocoa works best for which type of product. And therefore, new methods to help them to select this will be very nice because so far, people just have to do a tasting every morning in the factory. And then they decide more or less which is going where and which is used for which type of product. But as everybody knows, you get sick, for example, people leave the company, so lots of knowledge is leaving, and then you have an issue. And especially if there is, for example, a cold period and people have all the flu or whatever, then nobody is able to do this tasting anymore. And that's then for industry business a big issue nowadays. So having also instrumental methods to kind of support this process, that's really something that helps to deal with their daily business at least. So what we can do with the long-term prospects, I will give you a few more kind of bullet points in the outline to tell you where do we currently think that we might have a chance to change the situation and to at least partially prevent these effects. That chocolate will taste awfully in 2100 and at the same time being quite expensive. So that's probably something we cannot fully stop, but at least we are trying to make the production more sustainable and also giving in the end thereby a better way to also keep product quality. So let's have just a few bullet points. So before we go to that, I just wanted to mention this is of course just a very small part of the picture because we just looked now at what happens at the cocoa bean. So what happens at the plant level more or less. But this of course still far away from what you need to do to make it a real chocolate. That's also far away from what we did here for a very simple experiment. Because first of all, you need fermentation. This is usually done in the country of origin. And there you have, of course, many factors influencing the outcomes. And this is, again, something which makes the picture even more complex. But we, for example, think with our partners in our research network about maybe using certain strain to degrade compounds which are produced in the future at higher levels to a certain amount to limit their effect on the overall taste. So this might be an option that we use something like a specific starter cultures that we use in other products like cheese all the time. Maybe this is something we also will use in cocoa research and cocoa production in the future. So there is already, for example, work done by our partners in Switzerland, for example. They, for example, try to develop starter cultures to just help to avoid the development of fungi in these fermentations, because usually you don't want to have these molds growing on the cocoa. You just want to have the nice things which degrade a little bit the compounds in the cocoa and make the nice flavor precursors. But if there is a certain mold growing, then of course you have to throw it away more or less. And if you can prevent this by not spraying some fungicide on it, by just using maybe a different starter culture, that would be a nice and great sustainable way to improve this. And that's, for example, where people are already working in that area. And we think in the near future, it might be also used to tailor made cultures, for example, to improve the flavor, for example. This is then the fermentation. Next step is then real chocolate making. That's what's primarily done in industry, but also there we have different options to optimize processing, for example, to learn how we need to roast a cocoa which has more composition that we will see in the future. Maybe we need to change our roasting conditions to make it then less bitter, because if we roast at a lower level, it might lead to lower bitterness than we would get by using still the conditions we use nowadays. So that's also something important. And all this only works if you also have, of course, people like us who then have to analyze these hundreds and thousands of samples and also analyze the data. And also this is not ending at the level of research projects. We also work then with partners, for example, who develop apps on the phone who help then, for example, farmers in the country of origin to just scan the beans during the fermentation to understand, is this already well fermented, or should we wait another day until we stop this and try this? So also this is important, that we not use the data and all the tools we develop in research, that we also bring it back to the producers. And there, in those areas, for example, data science tools that can run nowadays on every smartphone more or less, and also use their cameras, for example, very nicely. This is something which has a great potential, and as you see, this is a very complex topic, and that's why we not work on this alone. As you see, there are many partners around Europe and the countries of origin involved to do this, and this is where we hope in the next future to even increase in the end the output of results from it. You have a question right now?
Chocolate, did you use plants from Africa, or did you use the indoor plants from Reading?
So for what you tasted right now, or?
In general, what is it? Because I just read like Ghent University and these curators in Belgium.
Yeah.
And they also have a project that was Space Bakery, where they do indoor meat.
Yeah, I know.
And so I was thinking, oh, that's interesting. Do they also use indoor plants?
So the problem is the yields from those plants in the greenhouse are way too low to really make chocolate out of it. So if you would ask in food industry how much they need to make a kind of pilot plant run, we talk about 100 kilo or at least 20 kilograms or so. So it's far away from what you can do, of course, from the greenhouse. So far, this is completely isolated. So the greenhouse is really to simulate conditions we start to simulate outside as well, but of course in a more controlled way. But everything that is used for real tasting, for changing processing conditions and so on, is coming from the field still. And this will also not change. But again, as we now have the tools to monitor, for example, what's the raw material quality which is going into these processes, we can, of course, already see differences between individual harvest seasons and so on. And we clearly see, for example, West Africa, the changes between individual seasons, whether it was a very rainy season and so on. So this is really something where we already by chance get those conditions sometimes and then can prove these models. And yeah, that's unfortunately a little bit the problem. We cannot produce enough in the greenhouse simply. Already the fermentation is a problem, because if you just have a few beans, this is not really a good model of what happens in the field. In the field, you have huge piles or boxes, and this climate you develop there with very special temperature and pH changes and so on. This is not really something we can model so far in the lab. So we try to do it on small scales, but still this is, for example, a gap between what the greenhouse can produce.
I also did a similar question because I might have just missed it, but did they also kind of try to simulate the precipitation changes or the rainfall changes?
Well, that's the part I skipped for today, but
Okay.
that's of course also what they do. And also looking just at two genotypes is of course just a small part of the picture. But in the end, this was something we started more or less for fun, by just having nice tools and the interest in doing research. But to do something on that level, of course, you need to look for funding then and to establish a larger network, which hopefully long time is then able to look into such a product. And also training people who will help industry in the transformation. And what we, for example, and you will see it on my last slide, about what we think can be done already nowadays in the field to kind of work on these issues. And one part of this is we will definitely need to improve the training of the farmers. They need to learn a lot of things which are currently, well, in principle, the knowledge is there, but not at the levels of the farmers and the people who really work on the plant. And this is something we also have in those projects in mind, that really people go there, train, improve the training, and so on, developing training materials. All this will be hopefully possible then in the near future. So let me just give you one part of the outcome tools is that we develop data science tools, as I mentioned. So we develop, for example, databases to help people to really share these complex results and make other researchers who are maybe just experts in the fermentation able to jump on board to see what do we already do, what do we already know, where might be a gap for additional contributions. So therefore, we develop then online tools and databases. And it's on the left-hand side, and on the right-hand side, you see, for example, a tool that we have developed, and that's a so-called flavor wheel. So that's what people do who do the central evaluation usually use. So they have here these different descriptors, so it's certain names of how you perceive certain things. And this is a big issue if you work in an international environment. Because in every country, you have slightly different translations for certain impressions. And we, for example, harmonize it that you can then select here just your language, and you can then move into a different language and get still the perfectly harmonized translation. So that also helps to improve, for example, the quality of data in this area, which is usually a big issue if you work with such large networks. And if you go back really to the cocoa in detail and think about what can we do, then the situation that we currently have is primarily driven by the problem that most of our cocoa is coming from West Africa. So if you look here at this plot, this purple region here is the production from Africa, especially Ghana, Ivory Coast, and then you see just a little bit, this green one is Asia, so that's improving now, but still, of course, just a small share, and then we have Middle and South America for the rest. And one part for where we see options to kind of stabilize the situation. Of course, this is something which is not helping necessarily the farmer in West Africa. For those, I will come to the second slide. But one option, of course, is definitely diversifying to not only relying anymore on South and West Africa, because there we have different regions where we really see on these climate models that the conditions will be very likely not perfect anymore for any cocoa production. And we also have there a high incidence for certain disease, which we don't see so far in other regions of the world. So that's one point that we think diversification should be here. The other point, of course, and that's maybe even more difficult to change, is just the economic system. Because most of the money that is made with cocoa is not ending up at the farmer, of course. It's lost somewhere in the commercial trade process, and even not necessarily at the manufacturers here than in Europe, for example. So really the step in between, the middleman in the trading process, this is really a huge share of the overall money. And if we could kind of change the system a little bit, at least, that more is in the end available to the farmers, they would then have, for example, in West Africa, the chance at least to look into things like pest management, which is usually simply too expensive for them, or also to train them to maybe replace older trees which are not that productive anymore, all these things. And therefore, I just have here certain lists. So the overall solution on the global level is, of course, the diversification that we see as a clear need to deal with these issues we have seen on the first slide, with this high price increase, with the loss in production we see in West Africa currently. But other points are, as I mentioned, available as well. So if we give to the farmers a bigger financial share, then this will enable to do them things like, for example, investing in more robust clones and so on. And especially the replacement of dated trees, which are currently used in these populations. This might improve definitely the situation in terms of productivity, and what I also mentioned already, the training of the farmers. So, for example, how to use fertilizers, which is in cocoa still something not systematically used, and in many regions it's simply not done at all. So we see, for example, we have a collaboration with a group in Israel who's also doing greenhouse experiments. They, for example, looked at what happens if you change levels of nitrogen, phosphorus, potassium, and so on. And they see sometimes dramatic changes in the productivity of the trees. And this would be, for example, something which definitely would help the situation of the farmers if they're able to do this and if they know how to do it, of course. Because just putting something on the field is, of course, also not the solution. So the fertilization, then pest prevention, of course, is something those farmers should hopefully learn in the future much better, how to prevent without having too much agents you need to spray and so on, certain diseases. Also, pruning of trees, for example, is a very important topic in cocoa, which is not used systematically usually. If you think about wine, for example, there's a whole science about how to do it, and people who are really making excellent wines spend really lots of time and labor on this. In cocoa, this is more or less not done systematically so far, but there's, we think, a huge potential to improve situation by, for example, training people to how to prune their trees correctly. Then also training people, what's the optimal harvest period? So currently there is sometimes more like a moon calendar or whatever to decide what is the right time. So it really is different on every village and every farm. And here we would improve dramatically the situation if people would be trained more systematically about, for example, the perfect harvest season. Also, for example, the fermentation and the drying. This is currently still done most of the time at the farm level, or at least at smaller collaborative centers, which are very close to the farm still. So definitely close to the origin and not in the big factories in the West. So that means also here we have the chance to improve the quality and also reduce, for example, losses due to wrong fermentations and drying. So also here we see a potential to, with simple training, improve the situation already. Then also, deforestation is an important topic. So of course, everybody knows what happens with the rainforest and so on, and taking away areas to just grow more and so on is definitely not the solution. And people are now trying to develop systems, planting systems and so on, where people really learn what best put together in the cocoa farm to get then a nice mixture which produces in the end stably a nice material. And also that we ideally can a little bit reduce the impact of the cocoa brokers on the overall system. And what we hope also with our research, besides the training to do, is of course coming up with maybe new clones that might help us to also mitigate some of the issues. But this of course will only work, as I showed you, with lots of research to understand the complex problems. And with that, I hope you had at least learned a little bit about cocoa. Unfortunately, I have no time machine to really show you what will be the taste of chocolate in 2100, but at least I think I showed you a little bit what will be something we need to deal with to really have something nice on our tongue in the near future. Okay, thanks.